Whenever disease causes multiple different types of symptoms, that disease is said to be a syndrome. Now, virtually all chromosomal abnormalities that arise in humans are syndromes, and that's because they cause multiple symptoms, both physical as well as mental symptoms. Now, the most common type of chromosomal abnormality in humans is Down syndrome, and this will be the focus of this lecture. So we're going to see that there are two forms of Down syndrome. The first form, the more common form, is trisomy 21, and the other form, the less common form, is translocation. So let's begin by focusing on trisomy 21. So the majority of the people in the world that have Down syndrome have the trisomy 21 form of Down syndrome. Now, what do we mean by trisomy 21? Well, trisomy means we have an extra copy of a chromosome, and 21 refers to chromosome 21. So remember, the karyotype of any normal individual describes 23 pairs of homologous chromosomes, and each pair normally consists of 2 individual chromosomes. So we have a total of 46 chromosomes in a normal karyotype. But if we examine the karyotype of an individual that has trisomy 21, let's suppose the individual is a male individual, this is what we're going to see. So notice every single one of these homologous pairs of chromosomes consists of 2 individual chromosomes except chromosome pair 21. So in an individual, in a male individual that has trisomy 21, they have an extra additional copy of chromosome 21. So we have not 2, but 3 of these individual chromosome 21s. And so that's why this condition is known as trisomy 21. So this is the karyotype that describes the karyotype of a, so this should be male individual who has Down syndrome. Notice that she has, or he has, an extra copy of chromosome 21, and hence this form of Down syndrome is said to be trisomy 21 Down syndrome. So the reason this is a male individual and not a female individual is because this is the X chromosome, sex chromosome, and this is the Y sex chromosome. If this was an X chromosome, then in that case, we would have a female individual. Now, let's discuss how the age of the female actually affects or actually increases the likelihood of Down syndrome. So remember, in female individuals, all the X cells that the female individual will ever have in her lifetime are formed before birth. So all the X cells a female individual will ever have are formed before birth. And what that means is, as the female individual actually ages, so do her X cells. And so as that individual ages, her X cells ages, and as the X cell age, the likelihood that nondisjunction will take place in meiosis and form aneuploid X cells increases. And so if the likelihood of nondisjunction increases, that means the likelihood of Down syndrome is also higher. So, although the occurrence of Down syndrome in the general population is actually very low, it's about 0.15%, it increases with the increase in the maternal age, in the age of that mother, for the reason that we just discussed. So recall that female individuals produce all their X cells before birth. So as the female individual ages, the X cells also age. And the greater the age of the X cells, the more likely that those X cells will actually undergo nondisjunction during meiosis. Now, to see exactly what we mean by that, let's take a look at the following diagram. So this is one example of how trisomy 21 can actually take place, can actually occur. So let's suppose we're dealing with a normal female individual. So since we're dealing with a normal female individual, that means each one of these pairs of chromosomes will consist of 2, and that includes chromosome pair 21. And because we're focusing on trisomy 21 in this diagram, I've only drawn this chromosome pair 21. So we have a normal female individual. So after interphase, we have the precursor X cell basically replicates each one of these two chromosomes. So remember, we have a normal individual, so we have one of these and one of these. Now we replicate it, and so we produce the sister chromatid that is identical to this chromatid, and we produce this second sister chromatid that is identical to this one. And so we have the pair of sister chromatids as shown in the following diagram. So this is after interphase takes place. So now let's fast forward to metaphase 1 of meiosis. This is what we're going to basically see. We're going to have the tetramers align along the equatorial plate. Technically, we have all these different chromosomes that also align, but once again, to save space and time, I've only included chromosome pair 21. So normally what happens is we have this mitotic spindle apparatus that forms, and these fibers extend from our mitotic spindle apparatus, and these fibers should attach onto each one of these respective chromosome pairs. And during anaphase 1 of meiosis, we have the segregation, the movement of these chromosome pairs to opposite poles. And so after meiosis 1 takes place, we form these two haploid cells. Now, because we're assuming no non-disjunction took place within this stage, we're going to have two normal haploid cells after meiosis 1. Now let's suppose, because the female's age is, let's say, relatively high, the chance of non-disjunction will increase. And so now let's suppose non-disjunction takes place within this cell, but this cell divides normally. So what happens is, within this particular cell, each of these fibers attaches onto each one of these respective identical sister chromatids. And so following cytokinesis after metaphase 2 of meiosis, we form the following two normal X cells. Notice that these two X cells are normal because they contain the proper number of chromosome 21. They contain one chromosome 21 each. But what happens in this case? Because the age of this X cell is high, the likelihood that non-disjunction takes place is also high. And so what that means is, what might happen is, this fiber doesn't actually attach onto this chromosome. This is the chromatid here. And so what happens is, this entire pair of sister chromatids basically segregates, moves to one side, and nothing moves to the other side. And so we form these two X cells, XL1 and XL2, which are abnormal. They have aneuploidy. One of these cells lacks a copy of chromosome 21, but the other cell has not one, but two copies of chromosome 21. And so now, if we take a sperm cell, so this is our sperm cell, that also has, so let's suppose we have a normal sperm cell, and that means we have a normal amount of chromosome number 1, we only have one. When this sperm cell, normal sperm cell, combines with X cell number 2, we're going to form a zygote that has the following genotype distribution for chromosome 21. So instead of having 2, as in a normal case, we have 3 copies. And this is what we call trisomy 21. So if a normal sperm cell, like the one shown here, that contains a single copy of chromosome 21, combines with X cell number 2 that contains an extra copy of chromosome 21, we're going to produce a zygote, an individual that has trisomy 21. Now, notice in this particular case, I've discussed non-disjunction taking place during anaphase 2 of meiosis, but non-disjunction can also take place during anaphase 1 of meiosis, and in both cases we can basically produce a zygote that contains trisomy 21. So trisomy 21 is the much more common case of Down syndrome. Now, the other much less common form of Down syndrome is translocation. Now, translocation, if you remember from our discussion on chromosomal abnormalities, translocation is the process by which a segment of a chromosome breaks off and moves onto and attaches onto a different non-homologous chromosome. So translocation can also lead to Down syndrome, but in an individual that has Down syndrome due to translocation, that individual actually contains the normal number of chromosomes, 46, but one of those chromosomes is actually abnormal. So not all Down syndrome cases are due to trisomy 21. Although the majority are due to trisomy 21, about 4% of the individuals with Down syndrome actually still have the normal number of chromosomes, 46, but one of them is abnormal as a result of the process we call chromosomal translocation. So, for example, let's suppose we have the following two normal pairs of chromosome. We have chromosome 14, and so this chromosome pair right here, and chromosome pair 21. Now, what do we mean by chromosomal translocation? So what chromosomal translocation means is a segment of one chromosome, so in this case, a segment of chromosome 21 basically detaches and moves on to this non-homologous chromosome. So remember, these are non-homologous chromosomes with respect to one another. So we have a breakage of a segment of that DNA take place here, and this basically moves on to this chromosome right over here. And so what we form is this larger abnormal chromosome, and the way that we call it is because we have a fusion of a segment of 21 onto 14, we call this chromosome 1421. So a segment of DNA on chromosome 21 can basically break off and reattach onto a non-homologous chromosome, chromosome 14. And the fused chromosome, this abnormal chromosome, is called chromosome 1421. So when the zygote basically gains this particular chromosome here, that individual, the zygote, still has 46 of these chromosomes, but the 14th one, this one, is abnormal because it contains this duplication, this duplicated portion of the gene, and that can also lead to the same symptoms of Down syndrome. So there are two forms of Down syndrome. The much more common case, the one that is affected by the age of that mother, is trisomy 21. And the less common case is translocation. This is the case where we still have 46 chromosomes, the normal number of chromosomes, but one of them, usually chromosome 14, is actually abnormal. It contains this extra segment of genetic information that leads to the same exact symptoms that we see in Down syndrome individuals.